And I will start recording. So when this pops up and says, like it's streaming, yeah, you can admit and blah, 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 all that stuff. All right. Hello, YouTube. Welcome to the Water, Wetlands and Watershed Seminar. Let me just make sure I'm recording to the cloud. And you may need to hit got it again, but you look at, okay. All right. Excellent. So it is a very wet day here in Gainesville. It is a perfect day for another water aquatic related seminar. So today we're very lucky to have with us Dr. Gretchen Lescord. Dr. Lescord is an assistant professor of applied limnology in the School of Forest, Fisheries, and Geomatic Sciences at the University of Florida. I think we'll hear a lot about today. She's also the director of a cool program called LakeWatch. So I won't say too much about it, except that it is one of the largest volunteer-based aquatic monitoring efforts in the entire world. Okay, Destin. Dr. Lescord received her PhD in boreal ecology with a specialization in stressed aquatic ecosystems. So that's why you came to Florida, right? We had a lot of stressed ones. And that was in 2019, and has studied chemical ecology and ecosystems all across North America. Her career goal, if I kind of taking some liberties here, but career goal to help provide the information needed to inform policy decisions and improve the management of lakes. I'm sure there's some other ecosystems as well, but trying to benefit freshwater ecosystems, not just for themselves, but for all of us that are using them. So with that, floor is yours, Dr. Lescord.
Thank you. That was a wonderful introduction.
I'll let people in from here.
Okay, great. Okay. Yeah, so you took all my buzzwords, which is great. Forestry, fisheries, and geomatics is a bit of a mouthful, but that's where I'm housed. I'm out at the Millhopper unit, so off campus normally, so it's nice to be on campus today. But what I'm going to talk to you about today has to do with LakeWatch. It's on public participation in water research and management, which has become a topic near and dear to my heart.
So now you just got to click one more time on the screen, and then we should be good to go. Okay.
Let's try that. Yeah, that worked. Whoop. Okay, there we go. So a bit of an outline of how I'm going to structure this talk. I'm going to start out by letting you know sort of what I study scientifically, set some context for the type of public participation that I do in my work. And then I'm going to talk really broadly about two forms or two categories that are very flexible categories of public participation, starting with community science, which for me has been largely rooted in Indigenous partnerships, which have some special considerations and really important aspects that we'll discuss. And then third, I'll go into citizen science, which is sort of the other category and where LakeWatch fits into, and I'll talk about more the program then. And then I'll wrap it all together and talk more about the future of participatory science in water research, what we're doing well, what we can improve on, how we can do it better, those sorts of things. And I just want to say that feedback's very welcome. I mean, I'm hoping that this sparks some interesting discussions here in the room, but also people online. These are my perspectives on some complex and nuanced things that are evolving. So a lot of this terminology and stuff is being debated right now. So if I spark something interesting, please chat with me after or send me an email. There we go. Okay, so as Dr. Coupland said, I'm a limnologist. So if we don't know that word, though it sounds like most people in this seminar do, but limnology is essentially the oceanography of inland water bodies. So both limnology and oceanography span a bunch of different topics, from ecology to biology, chemistry to physical morphology. And I've done work on all of those areas. I started out, like many of the students in my department, loving marine systems. I grew up on the Maine coast and thought I'd study marine ecosystems. But then for my master's, I went up to Resolute Nunavut, which is in the high Arctic, and I got to study 6 absolutely stunning freshwater ecosystems, one of which is Amatuk Lake, which is in the picture here. I got to catch these massive, beautiful Arctic char from these seemingly empty, clear waters, and the hook was set. I loved freshwater, and I never turned back. So I've been really lucky in my career. I've gotten to go and sample and work on lakes spanning all across North America. So up in that high Arctic region, and then I've done a lot of work in the boreal temperate zone. And now I moved here and I'm starting to learn a lot about our really unique subtropical ecosystems in Florida. Across all of them, I do what I've kind of called the new buzzword chemical ecology. So I use both chemistry and ecology to understand these ecosystems. That includes doing limnological modeling, water chemistry, and water clarity, aquatic ecology. So I use stable isotopes to understand food web structure and fish diet. And then I merge those two things together to understand pollution chemistry in these systems. So for example, I've done a lot of work on mercury biomagnification through aquatic food webs, PFAS, or those forever chemicals speciation in aquatic food webs and how fish diet influence it, arsenic speciation, things like that. So oftentimes I'm in a lab more so than in the field these days, but I definitely dabble in both. And I would say most, if not all, of my papers have fish in them. And there's multiple reasons for this. Fish are excellent environmental sentinels to tell us about pollution concerns and give us indications of water quality and ecosystem health, things like that. But also people really care about fish. So this has been sort of the conduit that has led me into public participation within science, is fish and the people who love them. And this is not a small topic in Florida. So in Florida alone, the freshwater fishery brings in 3.1 million anglers to our water every year. That results in $8.7 billion to our state economy and over 80,000 jobs. So this is not a small part of our lives here in Florida. Fish are very important, freshwater fish specifically. And who fishes in this room? Anyone? Who eats the fish they catch? Okay, eat fish. Well, for those online, I saw a few hands, but that's a very good thing. So most of the health agencies, the FDA, Health Canada, the WHO, all recommend we eat 2 meals of fish and seafood each week. And that's because they're low in the bad types of fats, high in the good types of fats. They're really good for our hearts. They're good for kids as they're growing up. And they have immense cultural, community and family importance for many groups and people. But wild-caught fish are also the main pathway for those environmental contaminants I talked about to enter the human body. So there's this balance of some really good pros and potentially some concerns as well. These concerns are elevated when we think about subsistence fishing. So most of my work to date has been done in remote areas of the north. And I work out of these small communities, many of which are indigenous. They don't have year-round road access, so they're only accessible by plane or by ice road. And that means that the people who live there have a hard time getting affordable, accessible produce. And so they rely heavily on the animals and plants that they can gather from their traditional homelands. So subsistence fishing is defined as fishing to feed yourself, your family, or your community, which is very different from going out and sport fishing, where if you caught a fish and you knew that it had a high level of mercury, you could toss it back in and stop at Publix on your way home. So this is not the situation I was dealing with in my early career. So it gave new importance to the work I was doing, and it led to a significant effort in community outreach. It started largely in field work in the boat as I was going out, because often we work with community members from these remote communities to collect our water and fish samples. And we talk about what I was finding, what they had questions about, what they weren't unsure about, what they thought was happening, and that would really inform my research. Later, I would present at things like land use planning meetings, which were held by the government, and all the leaders of northern communities were brought down, and I sat on panels and talked, kind of about these concerns more broadly. And then I've also done things like science cafes, youth programming, lots of media interviews, things that a lot of us do as well. But that was not community science, and I want to make that really, really clear. And I've learned that very well over the past couple of years as I started to really engage in community science. So this is a definition, a wordy definition from a nonprofit online, and I pulled out a few buzzwords to help us define what community science is for the purposes of this presentation. So instead of going into a community or into a group and saying, I have a question and I want to answer this, I go in and I say, this is who I am. This is what I know and can do. Like, this is my expertise. What do you want to know? What do you want to study? What are you worried about? What are your concerns? So we do the project centered around community priorities and helping to answer community questions. And we do it early. So we go in at the start of projects before we've designed things. And we get community input on how to do that design, where to sample, what do the results mean, helping us interpret those results and determine our conclusions. And then finally, the other point is that the goal of the project is first and foremost always to benefit that community. And something I'm really hoping to drive home with this talk is that does not negate our scientific outcomes that we need to produce. As researchers and academics, we need to publish, we need to present, and you can do both. We can do prioritize community needs, but still do those things as well. It doesn't mean to hinder it. So I'm going to give two examples of community projects that I'm currently working on. Both are partnered with Indigenous communities, and we'll talk a little bit more about that. These are really high-level overviews. I can't go through it in a ton of detail in the amount of time. But the first project is called Learning from Lake Sturgeon. We have an awesome webpage that my colleagues keep up to date. It's really engaging. If you Google Learning from Lake Sturgeon, it'll come up, or that QR code works. So I encourage you to check out our website. So the Learning from Lake Sturgeon is based in a river in northern Ontario. It was started in 2016 when Moose Cree First Nation had concerns about lake sturgeon in their traditional homeland. So a lot of hydropower operations were in part of their river system, and they were worried about these lake sturgeon moving and being able to live in these systems. So lake sturgeon are also known as Namao in Moose Cree, which is how we call them during this project. And that's my colleague from Moose Cree in the center there, Jennifer Simard, holding a lovely Namao just downstream of one of those hydroelectric dams. So the core of this project started out with telemetry tagging. So we would tag, you want to ask a question?
Yeah, just real quick, sorry to interrupt, but what's the language there on the top of that logo, the sturgeon logo?
Yeah, so those are syllabics in the Moose Cree dialect of Ojibwe. Yeah.
Sorry.
No, that's okay. No, questions are fine. So yeah, we started by telemetry tagging fish in the Matagami River, which is the one with the hydroelectric dams, and we compared their movement to a free-flowing river just next door to see the impact. This is a graphic from, we actually put together a graphical magazine about our project, which I brought a few copies for people to see if you want. There's also a PDF on our website, which is, it's pretty cool. It's a cool activity to be involved in. Since we started the project, we've sort of answered some questions, but then of course, many more have arose. And we've been able to get funding from the Department of Fisheries and Oceans, which is when I joined, as well as a number of other colleagues. And so now every Namao that we capture, we also, and we still tag them and do the acoustic data, but we also take blood samples for contaminants, fin clips for stable isotopes, which is my part. We run genetics on them. And then I have an awesome colleague at Ontario Tech, Danita Simmons, who takes mucus and runs omics analyses on them to look at stress hormones and things like that. So it's become this really wonderful multidisciplinary tight-knit group. You know, we meet every 2 weeks and chat about our data and our progression of our projects, and that includes Jennifer from Moose Cree, and we're getting some really good data from it. Other aspects that make this a community science project, from the very start, it was Moose Cree's concern, and we've been guided by Moose Cree's values and perspectives throughout. As I said, our group is a tight-knit. We meet frequently, and we hold great respect for their traditional knowledge that's shared with us, and we keep that in mind as we're interpreting our results and data. We get direct feedback and guidance from their Elders Advisory Council. They reviewed this outreach document for us and gave us some really valuable feedback just a few weeks ago. And we try and spend time in the community. That's another really, really important thing, whether that be during field work, but we also go out and join community gatherings. There's a really big one in the summer called the Gathering of Our People, or GOOP for short. And last year we had a booth there. This is the Royal We. I was not there, but Dr. Simmons had a great beading activity to show how omics techniques work to explain biomolecules to the community. So it's pretty cool. The second project I'll talk about is the First Nations Environmental Contaminant Project, or FNECP. That's actually our funder. I also call it the Wanapitei First Nation Project, which is the community partner, but they're actually the PI on this project. So that's a really important distinction. The grant and the money go to the community, and I am a scientific partner on this. So I am not the PI, I'm not the person in charge. My colleague at WFN is Sarah Lehman, so she's leading our group, and I am the environmental side of the scientific partner. And then I have a great collaborator, Brian Laird at UWaterloo, who's the public health side of this group. And then we have two great grad students. Taylor is pictured here. She just came down to visit to see Florida Lakes a few weeks ago and work on some of her R code. And then Amy has just started her PhD with Brian. So this project started, Sarah called me and said, there's this funding opportunity around First Nations and health. And a lot of people in the community have concerns about mining activities around their traditional homeland, and in particular, Lake Wanapitei, which is where the community is based. So she said, is there a way we can do a project on this? I'm studying metals and fish from the traditional homeland. And I said, yes, I can do the environmental side, but we need someone with more public health expertise. I called Brian, looped him in, and that's how our project took off. We're still in the middle of data analysis and things like that, so I'm not going to show you any data right now. What I am going to talk about, though, is just like I said, we went in early and asked the community what they wanted to study. And one of the really important things that came out is that when we think about eating a fish, we think fillets, right? But that's not the case in a lot of the rest of the world, actually. Other parts of fish are eaten, and in Wanapitei, the liver and the chum, which is also pyloric ceca, the intestines of the fish, are regularly eaten. So we knew that we had to sample both of those tissues as well. That was for the public health aspect. But what's happened is in Taylor's thesis, we're finding these really interesting patterns in the metals between those three tissues. We're actually finding some pretty novel research findings in that. So we're going to get a really great peer-reviewed publication out of this based on something that the community needed. So those are my two examples. Like I said, high level, and Indigenous community science is really unique under this umbrella of community science. So I do need to point these few things out. Indigenous communities are legal rights holders, and those legal rights are complex, and they're going to differ between Canada and the US. And second, there are significant historical experiences with many Indigenous people with Western science, and they're not positive experiences. So being aware of both of those things is really important when you're building these relationships, because they fundamentally change how we approach these types of projects. One of the aspects of community science is we always want to have a benefit to the community, and that is a challenging and complex topic when working with Indigenous communities. There's a great paper that just came out this year in Nature by Dr. Andrea Reed, who's at UBC. She runs a lab on Indigenous fisheries science, so I strongly recommend this paper for understanding some of the potential risks to the communities getting involved as scientific partners. One thing that our team has found really helpful when developing community relationships are research agreements and data sharing agreements. So basically that's a big long document with signatures at the end that details, what do you need? What do I need? How do we get there? What's the sensitive information that we're going to protect? What's the information that's okay to share? It also gives a mechanism for me to do the things that I need to do as part of my job, publish papers, present data, and things like that. It has a review mechanism that the community has input on how and when and where and why I do that to make sure I'm respecting their needs in the project. If you're interested in learning more about this, I strongly recommend FNIGC, the First Nations Information Governance Council, has an online course called The Fundamentals of OCAP, which stands for Ownership, Access, Control, and Possession. So I'd strongly recommend that course. I'll also do a quick shout out to the American Indian and Indigenous Studies program here at UF, which I believe the newest member of. We're a group of 18 or 19 faculty led by Ken Sassman in anthropology. And it's all of us work on or study some sort of Indigenous studies or science. One of the goals is to collaborate with communities and develop knowledge. And I like this last part, promote better understanding and respect among cultures. We're hosting an environmental seminar this fall. We're recruiting a speaker right now, an Indigenous scientist, to come in and talk about this. So if this is of interest, keep your eyes peeled for that seminar. Okay, so I'll quickly say there are many other types of community science that are possible. So I've been talking about, tribal organizations and communities, but there are other ways to do community science with other levels of government. Counties, townships, for example. HOAs are surprisingly engaged in this type of research, and other things, watershed alliances, hobby groups, student organizations. Does everyone have the Cornell bird app on your phone? Yeah, so now if you don't, you should check it out. It IDs birds by their calls, so I love using it in my backyard. That data goes to the bird lab at Cornell and is used. That's not community science, that's citizen science, which we'll get into, but birders are the best for data. Love birders. Okay, so that's a good transition into then what is citizen science. So again, for the purpose of this talk and sort of how I do my work, I look at citizen science using this definition from the National Park Service. Citizen science is when the scientists go in and say, hey, I have this cool question and I need your help in getting it answered and done. Usually that's because of a limitation on data or location access, things like that. And people who volunteer then help collect that data. Sometimes they help generating results or interpreting those results. And in turn, they have a connection to that science. They learn more about that specific topic, but they also learn about the scientific process overall. So it stops being this idea of science and starts being this activity of science, which is a very valuable leap. So there's no better example of citizen science than the Florida LakeWatch program. So that's what I'm going to talk about for the next couple slides. This is the former director of Florida LakeWatch, Mark Poirier, who just recently retired, and I took over from. So I want to acknowledge him because a lot of these early slides that I'm going to show were his successes, and I've learned a lot about Florida lakes from this man. So before I tell you about Florida LakeWatch, you need to understand how much water quality matters. So you probably know about all of our coastlines, and many in this room probably know about our freshwater resources too, but many people don't think about our freshwater in this lake. We have over 7,000 lakes and 1,700 rivers in the state. That's a lot of water. And nutrients are what control a lot of processes in those water bodies. And changes to nutrient loading from either human activities or natural activities can really change those processes within. Changes in nutrient can lead to differences in water clarity, invasive species, algae blooms, and fish kills, just to name a few things. And those have all been a problem in our state recently at some point or another. So we need to monitor them. We need to monitor nutrients and other water chemistry parameters so that if something is changing, we can mitigate if possible, especially if it's a human-caused reason. But how do you monitor 7,000 water bodies? The agency that does monitor this, the Florida Department of Environmental Protection, can only has so many resources and only is able to sample so many systems. So that's where the LakeWatch program comes in. So how we work is we train volunteers all across the state. So they usually live, work, or play on a water body and they care about it. And they go out on a monthly basis and take water samples for us. They drop them off in one of our collection centers, often at an IFAS extension office, and our regional coordinators go up and pick those samples up and bring them to our lab, which analyzes them for total nitrogen, total phosphorus, chlorophyll A, color, and conductivity. The data is then put through a data management system, QA/QC checked, things like that, and it's shared with all stakeholders who could be interested, academics, etc. But the most important stakeholder is the Florida DEP. So we give all of our data back to DEP so they can make informed choices about what gets monitored and things like that. We also give all the data back to our volunteers, and this is a really important part of our program. So we hold annual meetings across the state, 12 to 14 of them a year, where we bring all of our volunteers from a county or two together. We feed them, we give them a presentation on their region, we hand them an individualized lake report for their lake that they've been sampling. And then we have a really big Q&A, which can last hours sometimes, where they ask us questions and give us information about their lake that really does inform our research and our teaching practices. And then the whole cycle repeats every year. So the program to date, it started in the mid-'80s, and so far we've trained 1,800 volunteers across 6,000 sites and 2,000 water bodies. So that's resulted in almost 500,000 data points that's gone into countless peer-reviewed publications, student PCs, as well as information circulars, which I've brought copies of here, and I'll talk about again later on. So this is all possible thanks to the support of the Florida government. So in 1991, they recognized the importance of the program and they established a state statute that provides us support in this, the reason we're able to do this wonderful work. So this is what some of our data looks like. This is our total phosphorus concentration by year. This is from Lake Santa Fe, which was the first lake in the LakeWatch program. And Tom and Peggy Prevost were our first set of volunteers. They are still sampling for us. So this is just from a recent newsletter, we did a tribute to Tom and Peggy for their years of hard work with us. So that's a photo they sent us. So some of our volunteers have been with us for almost the entire time in the program, and they're incredibly dedicated. Tom and Peggy barely missed a month of sampling, so they love their lake. So this is our span. This map is pretty outdated. It's from 2015. It shows all the lakes that we sampled that year. We're working on an updated map right now, but I include it because I want you to see that we span everywhere from the tip of the Panhandle all the way down to the Florida Keys. So we have sites across the entire state. For these sites, most of them are lakes, about 65%, with another about 30% being coastal sites and rivers, and then a handful of springs and a few special projects make up that almost 500,000 data points. So LakeWatch has had a really big impact on both the research realm and limnology, but also on policy, also on how we manage aquatic ecosystems in Florida. And there are two key ways that this has been shown. First, that geology is the biggest driver of limnology in our state. So we have pretty variable geology. Some of our soils are really nutrient rich, like in the Bone Valley where we mine phosphorus. It's not too surprising that some of those lakes are green and have a lot of phosphorus in them. And then that leads to the second point, that eutrophic, which is such a bad term in so many news headlines, is not necessarily unnatural in our state. Some lakes are naturally, without any human influence, green here in Florida. For research, there's been a number of different projects on geology and water chemistry, but also on aquatic plants, looking at both native and invasive plants, doing massive surveys across the whole state. That's been baseline information for many studies moving forward. The same is true with fish populations. We have data, robust data on over 60 lakes across the state. They've also done special projects on harmful algal blooms and bacterial growth as needed, sort of as those issues have arose for our volunteers. And probably one of the best things that has ever come out of the program is this comparison that you mentioned earlier, showing that volunteer collected data is good data. So it's something we hear all the time when we say we work with the public, that, well, if it's volunteers, the data must be not good. But that is not the case. So Mark has done a lot of work, has published 3 papers on this. This is the most recent one. So on the y-axis here, we have the professional collected data, and on the x-axis is our LakeWatch volunteer collected data, and we see these nice, strong agreements between the two. We still do these comparisons as well. In the lab, DEP will go out and sample lakes and send us duplicate samples when we make sure that our analytical methods are in the line and we're getting the same results. So we do this twice a year, every year. So we're producing good data. So that was largely the history of the program. I am a new director, and so I'm coming in and we're calling some of the changes that have been occurring LakeWatch 2.0, at least that's how I've been calling it. This is a group photo of old and new, and it was in the retirement celebration of our most recent lab manager, Christy Forsberg, who just retired a few months ago. So I'm going to talk really briefly about sort of some of the changes and some of the things that aren't going to change, because when you take over a program that's working well, that's the first thing I did, was try and understand what is working well so that I don't screw that up and then see where we need to go. So we have 4 program objectives that we've sort of dusted off and refreshed a bit. The first 2 haven't changed much. Our core goal is to track long-term water quality across the state of Florida, to understand natural variations so that when anthropogenic disturbances come, we'll better be able to understand them to enable mitigation. A new one that I kind of revamped is researching both issues of emerging and long-standing concern that impact Florida ecosystems and beyond. And this is a recent photo of me with our new mercury analyzer in the lab that I'm very excited about, because this is where some of the pollution research will fit into, but also some continued research on things like harmful algal blooms and bacterial levels. And then the fourth one is relevant sort of for this talk, which is to bolster the role of citizen and community science in water research and management. This is what we look like sort of structurally. So we did restructure when we had the retirements and things like that. And we've divided duties into 3 core pillars. We have a manager for each pillar. So Marina Schwartz is our database manager. Dr. Joanna Lee is now our lab manager, and Dr. Liz Moreau is our new extension manager. We always had the lab extension and data activities, but they were sort of mixed between multiple individuals. And so kind of parsing them out has allowed us to accentuate some of those activities. In particular, pulling out the data and assigning someone to that role has really kind of opened up a lot of possibilities. So Marina has been doing a wonderful job. She's building large relational databases in SQL so that we can do some more enhanced and refined data QA/QC checks and really preserve it in the way we should be. There's a really good graphic, you know, Bart Simpson writing on the chalkboard. So there's a really great one that says Excel is not a database, and I almost put it in here, and I thought it might be a little triggering for some. But Excel is not a database when you have almost 500,000 data points. So we are building those good relational databases that will enhance our data export to DEP. It's also going to allow us to do things like build more interactive dashboards for our website. So this is a dashboard that was actually built by Alex Dew with FWC's Invasive Plant Management Board. He was interested in sort of writing some of this code and trying it out, and it's useful for his group to be able to see the LakeWatch data on a more spatial scale. So we're really excited to build on this and link this with our website and revamp some of that. And those databases are going to really help and enable that to happen. The other thing that I want to point out is I am also a teacher here at UF, and staff with the LakeWatch program has been involved in the course that I've taken over, which is Introduction to Fisheries Science and Fish and Limnology at the grad level, where we actually go out on Lake Alice and we do LakeWatch on Lake Alice. So we take water samples, but we also do fish population modeling. So they catch and tag bass with an electrofishing boat and get some really hands-on experience with the data. So I'm really lucky to have such an awesome undergrad course to teach and have the staff to come in and help me do it. And we resulted in a really long-term database on the bass population in Lake Alice as well, which has been published. That was Marina's master's thesis as well. But the other thing I'm doing with teaching is I mentioned at our annual meeting that we get questions from our volunteers. So what I've done is I've taken the most recent questions that I know have some literature behind them. And I've asked the students to pick one, and they have to conduct a thorough literature review on that topic. They have to write a full literature report, but they also have to present on it and make a plain language summary on it so that we can then go back to the volunteers and say, hey, we heard your question here. A student worked on this, and this is what they found in the literature.
Very efficient.
Pretty cool. It's going well. They're doing presentations right now. So. Okay, so I originally had way more slides about our newsletter and our information circulars. There's just not enough time. So a lot isn't changing. We have a lot of resources on our website that have worked really well. We're going to modernize some of them. Dr. Monroe is working on some of that. We're going to add some new ones, that sort of thing. But a lot of it is really functioning wonderfully. And it's such a powerful tool. So I really encourage you to Google us or go to lakewatch.ipis.ufl.edu. And stay in touch and see our future research there. Okay, so the final section on what's the future of participatory science and what are we looking at moving forward? So the first thing I want to acknowledge is word choice. So I have divided into two pots, citizen and community science, but I want to acknowledge that that's a conversation that's happening right now. And there are some places that are changing the term citizen science to community science. But I would really caution them to acknowledge that community science is really quite different in that it is led by community ideas and it is for the benefit of the community. Community science is hard. It takes time and it takes money. So calling something that's community science that isn't is not always the best choice. There are many other great terms. My favorite, I think, is participatory science, which is, I think, why I changed my title to this. But this paper here is that goes through and comparing and contrasting sort of what calls itself citizen science versus community science. It's a good resource. I had nothing to do with this paper, but yeah. Another great paper is Conrad et al. 2011, which I also had nothing to do with, but they go through some of the benefits and challenges of participatory science. Some of them are probably obvious, things like expanded geographic and temporal coverage, cost effectiveness, science literacy. On challenges side, funding, volunteer recruitment, those sorts of things are always going to be things that we're going to have to, it's an uphill battle. But there's been a lot of work done on those areas, how we do it well, how the programs that work, what are they doing and how do we mirror that? The things that we still need to do some work on the benefit side, is actually the outputs of the project. So what are the ecosystem impacts of these citizen science or participatory science activities? And what do the participants get out of it? How does LakeWatch change how you interact with your lake or how you see your water body, your access to your water body, those sorts of things that are really challenging to measure, which is why I haven't quite figured them out yet. On the challenges side, data accuracy, LakeWatch has been a leader in that space and we're going to continue to be because that's very important. And publishability and data utility is something I'm personally hoping to challenge because it can be worked around. So this is another view of the motivations for public involvement in research. And this is from McKinley et al. 27, again, nothing to do with that. Y-axis, we have the percent of programs, and then the x-axis, each bar is one of the motivations. So the bigger bars, the more programs do that. So that's things like educate the public, create long-term data, address a lack of data, educate youth. On the lower end of the scale, we've got attain data to affect a natural resource policy. And the very last one, only about 20% of programs answer a research question. Almost all of my publications have had community input, whether that be from those early fieldwork discussions that I told you about on the boat, and me trying to understand that ecosystem better with that community person, or from my community science projects. I have one paper in review looking at blood mercury levels in the Mayo right now. My other sturgeon colleagues just published a great paper on that movement data that's now out. I've got another paper drafted, 2 more being drafted, and many, many more in development. So we can publish this work and we can do it in a respectful and collaborative manner. This is, I think, my last slide, which is just to say that we are putting our heads together on the participatory benefit side. And this is something we're looking for colleagues on. So we've been talking with some great social scientists to help us develop surveys. One of the best things about LakeWatch volunteers is they fill out surveys, they answer them. They're excited to do it. So we can ask them questions about, how's LakeWatch influence your relationship with your water body? So I have to shout out to Dr. Monroe, our extension manager, for helping me think through this. And we're at early stages. And if anyone has any ideas or is interested in being involved with that, we would love to hear from you. Oh, I had one more slide, I lied to you. So take home messages, the big things. If you fell asleep, this is what I'm hoping you got from this talk. 1, freshwater ecosystems are just as interesting as marine ecosystems, and we should all keep working on them. 2, collaborating with the public enhances our science and helps refine our results. It's good all around. It does not hinder our progress. But 3, that participatory science takes significant time and effort. You say you can do things cheap, well, or fast, but you can only pick 2. You can never do all 3. And I think we'll all agree, we all want to do it well. So you either do it fast and well, and it's expensive, or you do it cheap and well, and it takes time. And that's been my experience. The wees I used are massive, massive royal wees. The picture up here is my sturgeon team. Picture down here is the Wanapate group, and this is my LakeWatch team. All those slides I showed are their successes, and I'm just thrilled to be a part of this program and be learning from them. So huge thank you to everyone on this slide, and thank you for your attention. I'd love to hear your questions, Katie.
Thanks so much, Gretchen. So we have actually quite a number of viewers on YouTube. So YouTube world, if you have questions for Dr. Lescord, go ahead and put them in the chat. Same with folks on Zoom. And we'll start in the room, and since it sometimes takes a moment. Oh, yes, thank you, Dr. Ewell. All right, so Kathy, please go ahead. You can unmute and ask a question.
Okay, thanks. Communities can be very diverse places, and I wonder what kinds of issues you face with people, often kids, who are playing pranks or just for whatever mental issues they have, how much you have to deal with those and how you do.
Yeah, thank you for that question. They are definitely complex groups to work in for very good reasons. The best example I can give is a very sad one, which is that in some northern communities, there was a number of youth suicides that happened when we had a really big segment of our project planned. And we knew we did not have to ask anyone. We canceled flights, we stopped. We knew that was not the time to go into the community and do work. So that's probably the most tangible example that I can give that really added to that time frame of that project, but was unquestionably the correct answer. So, yeah, I mean, communities are individuals as much as individuals are, so working with everyone is going to be different, which is why we do those research and data sharing agreements, why we have those in-depth conversations of what do you need, what is special about your community that might differ from our past experiences. So every one of these is going to be different, and my work with Moose Cree First Nation is very different from my work with Wanapitei First Nation because they need different things. So I don't have a glove that fits all hands for that answer, just a thank you for bringing that up because that is a huge part of this.
We have two comments from YouTube that are both more or less just hello, and you've got Andres Amota Gonzalez Sparta saying hello to you from the EU, and Dante Del Rosario, very much looking forward to how LakeWatch evolves under your leadership. So I don't know if you know those people, but they're saying hi.
Thank you.
So, you know, you talked a lot about like the data accuracy that that Mark and Dan have showed that the samples are good, right? And so, but then you have these long data records and you have many, many lakes. But I'm sure there are things you wish that were in those data sets. So you got TN, TP, conductivity, chlorophyll, second depth, right? What do you like? What is the biggest, I don't know, the top three things you're missing to be able to answer questions? What would you add? I mean, if you could add now. Reasonable, but you know, a little more money. What would you add now to be answering the questions that we have today about this?
Yeah, I think I'm going to answer this from my personal research interests, not necessarily from like what DEP needs. That is, I think we do what we need to address the numeric nutrient criteria and to help in that field. We're doing a good job. I have a really big interest in DOC and DOM quality, which is not something I know has been done across Florida lakes.
Here, those stand. What do those stand for?
So dissolved organic carbon, thank you, and dissolved organic matter. So little bits of carbon in the water, which can change drastically depending on what they came from and really influence biological activities, food web processes, mercury biodynamics, like things like that. So I have a really big interest in DOM and have some applications in to try and do some work on that. I'm also really interested in sulfate, which is not something that's been looked at extensively. So that is sometimes applied around our landscapes, and I think looking at sulfate and ecosystems would be something that I'd be interested in for things like mercury research and whatnot. So those are the two things that came to mind when you said that.
I wonder, do you get a lot of communities asking about things like Roundup?
Yes, we do, and so herbicides are a challenge and a concern on either side. I mean, managing aquatic invasive plants. So if you don't know, hydrilla is a big invasive plant. And if it is unmanaged, it will completely take over a lake like Orange Lake to the point where you can't get a boat through it. But on one side, it also is good habitat for fish and ducks. So hunters and fishers like a little bit of hydrilla, right? But then other people are really concerned about herbicide spraying. So, you know, there's been a lot of work showing that native plants can grow despite these herbicides. There's been a lot of good work to quell some of the fears. I am interested in doing some of that work to understand sort of some of the more future questions about these herbicides and things like that. But I recognize that it's a complex issue, and we do get a lot of questions on it. Yes, which differ greatly depending on
who's asking
the question.
Well, please, Linda, go ahead.
Yeah, I thought the sturgeon project was really interesting. Do you find that the local communities help you narrow down the fish that you're sampling, or do they happen to like request fish that you wouldn't normally think to see?
Yeah, for sure, that does happen. So same as the tissue experience that I brought up, that sometimes we get told they eat a certain fish that we may or may not be thinking about looking at. Oftentimes I'm scientifically more interested in the smaller fish because it's easier to relate things at the bottom of the food web to what's happening in the water than the top of the food web because you're several steps away sort of in terms of nutrients and things. But a lot of times people go for the big fish at the top of the food web. So sometimes when I'm talking to sport fishermen, it's that dynamic. But in communities, sometimes they are more interested in those small fish, which they do harvest. In the Moose Cree project, right now we're talking about potentially doing some other fish species, but we've been really focused on Nemeo for that work so far because, I mean, in the North French, I can't remember the exact population, but there's like 30 something sturgeon in the river. So like they're an endangered species, and the Moose Cree homeland is one of their last strongholds. So that's a huge conservation question as well. Thanks.
Yeah, I'm curious to know about something, and but we know that fish are indicators of water quality. And I would like to relate it to your experience as a limnologist and then the experience of anglers. And in one of the slides you showed us the bass. I want to know if you catch a fish and then you do not take it to the lab. Based on your experience, can you tell that this fish is contaminated or not? Or like, or have you talked to the anglers because they are doing this for many years? If you see different two water bodies, you can say that this one is of good quality, this one is not of good quality. What about the fish in those two water bodies?
That's such a cool question. The short answer is no. I need data. So what came to mind when you asked that was mercury, and there's no way to tell if a fish, how much mercury a fish has without analyzing it with some pretty refined analytical techniques. Same for all the other contaminants coming in my head. What you would be able to tell is things like bacterial infections or like skin infections on the outside. And then the data that we do get is on size and growth. So in one lake, those bass might have higher growth rates for one reason or another. Maybe there's ample food there because there's more nutrients, or maybe there's not. So we can tell some of that by not taking samples from those fish, by just looking at their lengths and weights and things like that. But for most of what I showed, I've always had to take samples and run some analyses, yeah. But I mean, in terms of growth and weight, that's the type of thing that a local angler can tell you where to catch the big bass. I mean, that's why fishermen don't like telling us where they fish, right? So for sure, they know where the bass are big, absolutely.
Okay, thank you.
Yeah.
So I guess my question is sort of how all this gets started in the sense of who seeks who in a lot of these, more the community science relationships I'm thinking are these First Nations people, communities reaching out to scientists saying we have this issue. Are you all going out and finding ambassadors? Like how does these things get started, especially like the political climate, thinking more about there's definitely people who are not particularly trustful of scientists and researchers at the moment who could potentially be incredibly good resources of this kind of data that would be useful to them. I'm thinking about hunters and stuff like that outside of water, but just on the broader scale, how do you get these relationships started?
Yeah, the end of your question there were all amazing points that I do not have answers to, other than time and exposure, experience, shared experience, like going there and talking with people. But how they get started, again, just like the first question, it's very individual, it's very community. They may have a specific question, so one in particular called me because they knew of my work. It was at nearby the university where I used to work. So in that case, that was launched that way. In other situations, we can go in and say, hey, there's this money and I can do this. Are you interested? And sometimes you hear no, and you need to respect those no's. And sometimes you hear yes, but I want to do it this way. And then you kind of have to pivot. So it can go a bunch of different ways. And that's true with Indigenous communities, which have many of those, considerations in terms of past experience. But it's true with any community group. You can go to an HOA and say, I want to study this herbicide. They could say, I don't care about that herbicide. I want to know about this one. And then I got to find a whole new lab to do the analysis in, So I guess you have to remain flexible and be able to pivot, but also have those conversations early and often is what I would say is the only way I can sum that up across community groups. Good question. Thank you.
We have one more from YouTube. This is from Andres Gonzalez Zapata. He asked, do you know about low-cost sensors for water chemical monitoring, like pH or turbidity sensors? Can you recommend some resources about that aspect for monitoring? Probably not in 1 minute, but yeah.
For a colleague, and I ended up falling down a rabbit hole of finding out that, I mean, in general, institute monitors are not usually as good as taking samples and analyzing them in a lab, but I'm sure there are pH ones that work perfectly well. Yeah. I'm not sure. Please email me and we can talk about this. My team is also amazing with these types of questions, so they might have some input, but nothing off the top of my head. I'm sorry. Okay, Andres, so you got the invitation to email. Okay, great. All right, any last questions? Okay, great. All right, let's give another round of applause to Dr. Vesco. Thank you so much. Thank you, you too. We will see everyone next week. Never quite have this ready. Next week, we are going
Be here.
That should
So
be.
you better be here.
Bad next week.
Yeah, so hopefully we'll have better weather next week. And we will be hearing from Patrick Inglet.
Yeah,
So Professor
excellent.
Patrick Inglet in Soil and Water Sciences. He has given us TBA for his title, so